The science behind modern point-of-care biosensing. Why point-of-care diagnostics matter. Point-of-care, or POC diagnostics, refers to medical tests done outside traditional laboratories, usually at or near the patient. This means testing can take place in hospitals, clinics, or even at home. The goal of POC diagnostics is to provide rapid, accurate, and cost-effective results. Instead of sending samples to a central lab and waiting hours or days, results can be available much faster. Advances in biotechnology, electrochemistry, optics, electronics, and materials science have helped move testing from laboratory benches to portable, point-of-care systems. Global infectious disease outbreaks have clearly shown the need for rapid detection tools. The demand for convenient diagnosis, monitoring, and screening has grown greatly, making POC diagnostics an important part of modern healthcare. What is a biosensor? A biosensor is a bioanalytical tool that detects a biological or biochemical response by converting a biochemical signal into an electrical signal. Every biosensor has four main components. First, the analyte. This is the molecule we want to detect. It can be small molecules, ions, viruses, bacteria, proteins, nucleic acids, or even whole human cells. When selecting an analyte, four conditions must be considered. It must have a clear link to the disease of interest. It should be present at a sufficient concentration for measurement. Its concentration changes should reflect a physiologically relevant parameter, and there must be a known selective recognition element for it. Second, the recognition element. This part interacts specifically with the analyte. It can be protein-based, such as enzymes or antibodies, nucleic acid-based, such as DNA or RNA, or lab-synthesized, such as nanoenzymes or molecularly imprinted polymers. Third, the transducer. This converts the biochemical interaction into a measurable signal. The signal may be electrochemical, optical, thermal, acoustic, or another form. Finally, data handling procedures manage measurement, denoising, deconvolution, correlation, and interpretation of the signal. These four components together explain how a biosensor works. How biosensors recognize targets. Recognition elements determine how the biosensor identifies its target. In enzyme-based biosensors, the enzyme catalyzes a reaction with the analyte. For example, glucose oxidase catalyzes the oxidation of glucose to glucolactone and produces hydrogen peroxide, or H2O2, as a byproduct. The amount of H2O2 directly relates to glucose concentration. However, enzymes are sensitive to temperature and humidity, so calibration is necessary to maintain accuracy. Immunosensors rely on antibody-antigen interactions. The binding between an antibody and its specific antigen creates a detectable signal. These systems may face challenges such as cross-reactivity, non-specific binding, and difficulty detecting very low analyte concentrations. DNA and RNA biosensors work through complementary strand binding. A nucleic acid sequence binds specifically to its complementary sequence. This allows detection of viral RNA or genetic mutations such as BRCA1, BRCA2, or KRAS mutations. Molecularly imprinted polymers, or MIPs, are lab-synthesized materials. They are created in four steps: selecting a template molecule, polymerization around the template, removing the template, and then allowing selective rebinding of the target molecule. The polymer forms cavities that are complementary in size, shape, and functional groups to the target. MIPs are more stable than biological receptors, but they require rigorous validation and regulatory approval. How signals are converted. After recognition, the interaction must be converted into a measurable signal through transduction. Electrochemical biosensors detect changes in electrical properties. Amperometric sensors measure current produced by an electrochemical reaction. Potentiometric sensors measure changes in voltage. Impedometric sensors detect changes in impedance when the analyte binds. Optical biosensors use light. Surface plasmon resonance measures changes in refractive index near the sensor surface. Fluorescence measures changes in fluorescence intensity. Absorption spectroscopy measures changes in light absorption. Piezoelectric biosensors use the piezoelectric effect. When an analyte binds to the sensor surface, it adds mass. This causes a shift in resonant frequency. The frequency shift is proportional to the mass change. All these methods can be affected by environmental interference, which may reduce reliability and sensitivity. Modern portable platforms. Modern biosensing technologies combine these principles into compact systems. Lab-on-a-chip devices use microfluidics to handle very small volumes of liquids, from nanoliters to microliters. They can combine sample preparation, detection, and analysis on a single chip. Lab-on-a-disc systems use centrifugal force from a spinning disc to move fluids through microchannels. This allows automated mixing, separation, and detection. Wearable and implantable sensors allow continuous monitoring. Many of these devices can send data wirelessly for real-time health tracking. Paper-based biosensors use capillary action to move samples along a paper strip, often producing a colorimetric response. These systems are low-cost, portable, and disposable. Together, these technologies provide miniaturization, integration, portability, and real-time diagnostics. Applications and limitations. The World Health Organization defined ideal point-of-care tests using the acronym ASSURED: affordable, sensitive, specific, user-friendly, rapid, equipment-free, and delivered. POC biosensors are used in bedside clinical care, at-home care, and public health monitoring. Examples include glucose meters and pregnancy tests used at home, as well as environmental and food safety monitoring. However, POC technologies often do not reach the same accuracy and reliability as traditional laboratory tests. They may have lower sensitivity, higher background noise, and usually detect only a single analyte. Environmental factors, such as temperature and humidity, can also affect performance. It is important to understand these limitations to keep scientific credibility. Artificial intelligence-assisted biosensing. Artificial intelligence can improve biosensing at many stages. AI-assisted POC biosensing includes identifying a problem, collecting relevant information, training a model using algorithms, and applying the model. Machine learning and deep learning can analyze complex signals, extract patterns, classify data, and improve diagnostic accuracy. For example, AI can analyze continuous glucose data to predict hypoglycemic or hyperglycemic events. Machine learning models have also reached up to 89% predictive accuracy in mental fatigue analysis. AI can improve transduction by predicting plasmonic nanostructure geometries that give optimal responses. It has also enabled signal fingerprint detection of the P16 protein down to 0.6 picograms per milliliter. In recognition element design, tools such as AlphaFold 2 have predicted over 200 million protein structures. RosateaFold uses a 3-track neural network to achieve similar structure prediction accuracy. AI also supports omics-driven biomarker discovery and multi-analyte analysis. For example, machine learning has improved Lyme disease diagnosis using paper-based biosensors. However, many AI models function as a black box, which creates challenges in clinical settings. Future of next-generation POC devices. The next generation of POC biosensing devices aims to achieve assured certification worldwide while maintaining accuracy comparable to laboratory-based tests. Future devices must be easy to use, non-invasive, cost-effective, and patient-friendly. Because biosensors collect and transmit sensitive health data, data privacy and cybersecurity are major concerns. Regulatory frameworks must adapt to allow safe and faster adoption of new technologies. Integration of AI and machine learning will be essential to improve sensitivity, optimize performance, and interpret complex biomarker data. Multiplexed biosensing will allow detection of multiple analytes at the same time. Sustainable and environmentally friendly materials will help reduce waste and support green healthcare technologies. Overall, point-of-care biosensing brings together biology, engineering, and artificial intelligence to make diagnostics more accessible, reliable, and responsive to global health needs.